Which of the following is not a characteristic of the peptide bond? Not. Okay, so it has a partial double bond character. Yes, peptide bond has a partial double bond character. It is usually found in the trans conformation. Yes, that is also correct, because at the peptide bond the position of the oxygen and the hydrogen is opposite side of the peptide bond, so it is in the trans configurations. C, it is highly flexible and rotates freely. No, it is not flexible, it is a rigid bond, so it is not the characteristic of the peptide bond. So here the correct answer will be C. Let's see other one. It is a planar due to resonance. Yes, that is also correct, but we need to choose which is not the characteristic of the peptide bond. So here the correct answer is C. It is highly flexible and rotates freely. Next, what are the two dihedral angles plotted in a Ramachandran plot? Dihedral angles are the angle between the C alpha in N and C alpha in C, and it is phi and psi. So here the correct answer is A, that is phi and psi. Omega. Omega is the angle present between the peptide bond between the C and N, but it is not plotted on the Ramachandran plot. D is also not the correct answer. So here the correct answer is A, phi and psi. Which amino acid has the most restricted allowed regions in the Ramachandran plot? In the Ramachandran plots, there are the two special cases which is not found in the stable secondary structures like alpha helix and beta pleated sheet. These are the proline and the glycine. While the proline has the most restricted regions, and it is generally present in the fourth quadrant, because in the proline the cyclic ring structure is present, and because of that the amide hydrogen is not available to form the hydrogen bond which is essential for the to form the secondary structure. Therefore here the correct answer is the proline, while the glycine has the most allowed regions, because in the side chain only hydrogen is present, but here is the most restricted, so the answer is A, proline. Which statement about the peptide bond resonance is false? We need to choose incorrect one. The CN bond has a partial double bond character. Yes, it is correct. Resonance leads to the planarity of the peptide bond. This is also correct. Peptide bond rotation occurs freely due to resonance. No, it is a rigid bond, so rotation cannot take place freely. And the last is the oxygen carries a partial negative charge due to resonance. Yes, this is also correct, but we need to choose the incorrect one, so answer is C. Ramachandran plot is used to analyze. So Ramachandran plots, we had learned that it is about the protein secondary structure. So let's see, DNA structure, no. RNA folding, no. Protein secondary structure conformation, yes. And carbohydrate configuration, no. Here the correct answer is C, protein secondary structure conformations. Which type of structure falls into the upper left quadrant of the Ramachandran plot? Out of 4 quadrants of the Ramachandran plot, the upper left is for the secondary structures like the beta sheet, antiparallel, parallel, beta sheet. Upper right is for the left-handed alpha helix. Bottom left is for the right-handed alpha helix, and the fourth is the disallowed region, last quadrant. Here question is asked about the upper left, so the answer is C, beta sheets. Which amino acid is least restricted in the Ramachandran plot? Least restricted means most allowed regions. So glycine, in the side chain of the glycine, it has the shortest side chain, only hydrogen is present. Only because of only the hydrogen is present, so least steric hindrance. Least steric hindrance means most allowed regions and the least restrictions on the Ramachandran plot. So here the answer is B, glycine. Which enzyme catalyzes peptide bond formation during protein synthesis? Peptide bond formation takes place during the process of translation. Okay, and the enzyme involved here is a peptidyl transferase. So others are not the correct answer. DNA polymerase, RNA polymerase, and the ligase is not the correct answer. Here the name of the enzyme involved in the peptide bond formation is peptidyl transferase. So correct answer is C. Who developed the Ramachandran plot? So just by looking at the options, we can say that by looking at the name, it is Sir G. N. Ramachandran who developed the Ramachandran plot. So here the correct answer is A. Which atoms form the planar structure of the peptide bond? One peptide bond is formed between two amino acids. So of the two amino acids C alpha and C alpha will present at the corner, and the peptide bond is formed by a carbonyl oxygen of one amino acid and amide hydrogen of the second. So CONH will be present at the center. So total these 6 atoms are present which form the planar structure. These are C alpha, CONH, C alpha, and the correct answer is A. How many atoms lie in the plane of the peptide bond? So just we had discussed the atoms which form the planar structure of the peptide bond are C alpha, CONH, C alpha. So these are total 6 atoms. So here the correct answer is C, 6. The Ramachandran plot helps in identifying, these are the following options. So in the primary structure of the proteins, when it undergo the folding, it forms the secondary structures. So here the dihedral angles are present between the C alpha in N and the C alpha in the C. So particular combination of the dihedral angles will lead to the one stable secondary structures, but not all the possible combination of the phi and the psi are allowed due to the steric hindrance of the side chains. So this is explained in the Ramachandran plots which combination of the phi and psi are allowed which form the secondary structures and which is not allowed. So here the correct answer is A, allowed and the disallowed dihedral angles in proteins. Which region of the Ramachandran plot is commonly occupied by right-handed alpha helices? Alpha helices are of two types, left-handed alpha helix and the right-handed alpha helix. So there is a trick to remember it. One is present at the top, another is present at the bottom. So it is opposite of the region which we are talking. For example, if it is a top right, so right region, so left-handed alpha helix is present. Another at the bottom, but bottom left, so right-handed alpha helix. It is opposite of the region. Okay, so here the right-handed alpha helix, so answer is bottom left quadrant. Which of the following dihedral angles phi and psi is most favorable for the beta sheet? Beta sheet structure lies in the first quadrant or the top left regions of the Ramachandran plot. In the first regions, the values of the phi and psi will be in the minus plus, where the minus and the plus sign is for the directions. So here the answer is minus 120 degree and the plus 135. So answer is C. Which structural element is found in the top right quadrant of the Ramachandran plot? So just we had discussed alpha helices. There are two types, left-handed alpha helix and the right-handed alpha helix. One present at the top, another at the bottom, and the direction is opposite of the regions. So if it is a top right, so left-handed alpha helix, bottom left, so then it is a right-handed alpha helix. Here the question is asked about the top right. Therefore the answer is B, left-handed alpha helix. Which experimental technique is commonly used to validate Ramachandran plot? Here answer is A, X-ray crystallography. Which amino acid is known as helix breaker? We know the two special cases, proline and the glycine, where the proline has the most restricted regions on the Ramachandran plot because of the cyclic ring structure, that amide hydrogen is not available to form the secondary structure. Therefore, it produces a sharp bend or the kink where it is present. Therefore, the proline is known as a helix breaker. So, answer is C, proline. Why proline is considered a helix breaker? This we had just discussed in the last question, that due to the cyclic ring structure, it lacks a free amide hydrogen for hydrogen bonding. Therefore, it forms a sharp bend or the kink where it is present. Therefore, the proline is considered as a helix breaker. So, answer is B. How do proline and glycine affect alpha helices? So, we will see one by one. Proline, as the cyclic ring structure is present, therefore, amide hydrogen is not available to form this hydrogen bond which is essential for the secondary structures. So therefore proline is a helix breaker, while the glycine in the side chain only the hydrogen is present. Therefore it will have the least steric hindrance and it produces a higher flexibility. So because of the higher flexibility, it will destabilize the structure. So here the correct answer is B. Proline is helix breaker while the glycine destabilizes due to its flexibility. Answer is B. Which structural motif commonly contains proline and glycine. Proline, it is a helix breaker, while the glycine, because of its higher flexibility, it destabilizes the structures. So these are not generally present in the common secondary structures like the alpha helix and the beta pleated sheet, but proline and glycine, it is present in the beta turn. So here the correct answer is B, beta turn. What are the typical phi and psi angle values for an alpha helix? Here it is not mentioned whether it is a left-handed or the right-handed, so we'll consider the right-handed because that is the most common one. It is lies in the bottom left, which has both the negative value and the value is a minus 60 degree and minus 45 degree. So here the correct answer is A, phi minus 60 and the psi minus 45 degree. Which bond in the polypeptide backbone does not have free rotation? C alpha and N and the C alpha and C have the free rotations, and we had discussed there the angles are the phi and psi. Peptide bond, due to the partial double bond character, it is a rigid, therefore it cannot rotate freely. So here the correct answer is C, peptide bond, it does not have the free rotation. Answer C. The psi angle represent rotation around which bond? So psi angle is present between the C alpha and C. So there is a trick to remember C, C and psi. Okay, so that is how we can remember it. So psi angle present between the C alpha and C. Correct answer B. The phi angle represent rotation around which bond? Just we had discussed the psi angle present between the C alpha and C. So C, C, psi. If we remember one thing, we can remember the other thing. So other between the N and C alpha. So phi angle present between the N and C alpha, and the correct answer is A. Which bond in the peptide backbone is associated with omega angle? Omega angle is present between the peptide bond. N and C alpha is with the phi, C alpha and C is with the psi, and the CN means peptide bond has a omega angle. So here the correct answer is C, CN peptide bond. What is the typical value of the omega angle in the most peptide bond? Omega angle is present between the CN, that is a peptide bond. Peptide bond, it is a rigid bond, it does not rotate freely, and as it is a rigid bond, it generally has a angle 180 degree. So here the correct answer is C, 180 degree. Thank you for watching and subscribe NitVed Bioscience for more videos. Thank you.